morning children and welcome to this video of revision. Today I will take the topic of biotechnology. As some of you had expressed your desire to discuss the principles and the application of biology with you. First today in this video I will take the principles of biotechnology. There are two principles of biotechnology. First is genetic engineering. What is genetic engineering? We keep hearing about this term very often. It is the technique to alter the chemistry of genetic material. It can be DNA or RNA to introduce these into host organism and thus change the phenotype of the host organism. So the first principle of the biotechnology is genetic engineering, that is the alteration of the chemistry of genetic material. By now all of you know that genetic material is DNA. And then after changing it, altering it, again inserting it into the host organism. The second principle is maintenance of a sterile ambience. That means to create an atmosphere which is free of microbial infection and to enable the growth of only desired microbes or eukaryotic cells. Whether they are microbes or eukaryotic cells, they should be without any infection. They should develop in a nutrient sterile atmosphere. And the production of biotechnological products like antibiotics, vaccines, enzymes, etc. is our aim of biotechnology. The most important field of biotechnology is genetic engineering or recombinant DNA technology. Now, through this, we have gone into the field of cloning, gene banking, formation of transgenic organisms, both in plant field and in the field of animals. These are based on rDNA technology. R means recombinant DNA technology, directly or indirectly. Now, what is recombinant DNA technology? How we can change the DNA? How we can get the genes of our own interest or desired genes? For this, there are some steps, and today we will see those steps. So we have seen the two principles of biotechnology. One was genetic engineering and the second is maintenance of sterile ambience. Now we come to the steps of rDNA or recombinant steps of recombinant DNA technology. So in this, the basic steps. The first step is the gene which has to be cloned. The gene that you want to make many copies of it, that is to be isolated from the genome of concerned organism. For example, if we want to make insulin, so we have to extract out or isolate the genes responsible for the formation of insulin from some organism which is having the good quality and insulin in abundance. So that is the gene of mammal we will take. The second is, so the first thing is to isolate the genes of interest from the genome of concerned organism. Second is a suitable plasmid or vector is isolated from a bacterial cell. Now you know what is a plasmid. Plasmid is a part of bacterial cell. Let's say here the bacterial cell is E. coli, and that plasmid carries additional chromosomal or hereditary information. And vector, what is the use of vector? Vector will help in transferring this gene to the host organism. So we have taken out the plasmid. Now you know the plasmid is generally circular in structure. So the third step, first was taking out the gene, isolation of gene. Second was isolation of the plasmid or vector. This plasmid will work as a vector or as a carrier. And the third step is to cut the plasmid with restriction enzyme, or when we will cut it, it will become straight. It becomes straight. Restriction enzyme, this opens up. This straightening opens up the cloning sites in the plasmid DNA. And the circular plasmid becomes straight. The desired gene is also cut by using the same restriction enzyme. Restriction enzymes, they are also called molecular scissors. So they are cut with the same restriction enzyme is used for cutting the plasmid and the desired gene. So this was the third step. Now the fourth step is now the desired isolated gene, that is insulin, that we had taken from the desired organism, that is mammal, is inserted into this plasmid with the help of enzyme ligase. With the help of enzyme ligase will help in joining, in inserting the desired gene into the plasmid. Now, as a result of joining of DNA into the plasmid, desired DNA into the plasmid, now the DNA has changed. It is called recombinant DNA. It is no more the original type, but now it has become recombinant DNA. Or another word is chimeric. It is also called chimeric DNA. Then what happens? Now the recombinant DNA is reinserted into the bacterial cell. Here, as I told you, the bacterial cell that we had taken was E. coli. It was E. coli. So we will transfer this rDNA into bacterial cell, that is E. coli, and the bacterial cell, because of recombinant DNA, is getting transformed. It will get transformed. Then comes the next step. And the next step, so the fifth step was the joining of the gene into the DNA. Sixth was putting back the recombinant DNA into the bacterial cell. And now comes the seventh. Seventh step is the transformed cell. Because of the insertion of the rDNA into the bacterial cell, the phenotype or the characters of the bacterial cell have got transformed. And in the seventh step, the transformed cell is now grown in a nutritional medium. It is kept in a nutritional culture medium, and this facilitates the replication or the division of the bacterial cell. And with each division, the new copies of this recombinant or the transformed bacterial cells are formed. New copies which are transformed are formed, which has got insulin genes in it. Then last is cell is allowed to carry on the expression of inserted gene, and thus a large amount of insulin is produced. Now, when the many copies are formed, so naturally a good amount of insulin is formed. So I hope these steps are clear to you. Very simply, we just have to go step by step. First step was that isolation of the desired gene. Here we have taken the example of insulin gene. The genes which were responsible for the production of insulin. Second, isolation of plasmid from the host or vector, that is plasmid, and then third was cutting off the circular plasmid with the help of restriction enzyme, making it straight. Then next step was cutting off the gene also with the same restriction enzyme and combining both of them, the desired gene and the plasmid. This is done with the help of ligase, and this gives us a new form of DNA, which is now called recombinant DNA. Then inserting this recombinant DNA into the bacterial cell, and the whole picture of the bacterial cell will change. We say now the bacterial cell is recombinant bacterial cell. Then putting this bacterial cell, transformed bacterial cell into a nutritional culture medium, which will facilitate the cell division and the formation of the bacterial cell, and in this way we will get many more copies of bacterial cell. So this is how the whole process of bacterial cell takes place. Now we said one thing, that was isolation of desired gene. This is also known as gel electrophoresis. The method that is used for taking out the desired gene is known as gel electrophoresis or isolation of desired How this is done, this we will do in the next video. So please go through these videos and revise with me.